Good afternoon and welcome to today's EMSL LEARN webinar, where we will be talking about our capabilities for chemical and biological characterization of critical minerals and metals. Information about our next webinar will be provided through our social media channels and via email. This is a great opportunity for you to subscribe to our newsletter if you don't already, so that you can be informed about upcoming webinars and also about our events, which does include the EMSL Summer School, which is coming up July 7th through the 11th, where we'll be focusing on fundamentals of artificial intelligence and machine learning, large language models, and the use of those models to create semi-autonomous software agents. But for today, let's focus on our three speakers. First up, we're going to have Shuttha Shutthanandan, who's going to walk you through how X-ray photoelectron spectroscopy can be used to determine the chemical and organic speciation of compositions on mineral surfaces. Will Q will follow him with a discussion on how FTICR mass spectrometry can be used to explore the properties of metal-binding molecules such as metabolites and proteins. And closing out the webinar, we'll have David Hoyt, who will demonstrate how liquid-state nuclear magnetic resonance and its tunable probes can be used to provide spectra-rich information on chemical functionality from mixtures on pure compounds. The last 15 minutes of our webinar today will be reserved for a question-answer session. We invite you to post your questions for our speakers in the chat. We'll compile those and have our speakers answer those at that time. And with that, I'd like to pass the webinar to Shuttha.
Morning, Heather. Heather, can you send me the link again, please? Can you guys hear me?
Yes, we can hear you. Go ahead, Shuttha.
It's not moving though.
Okay, we'll go ahead and advance.
Okay, thank you. So you guys have to advance, it looks like. Yeah. Thank you. So everyone knows there is a growing and strong interest recently in rare earth elements and critical minerals, because these minerals are essential for modern technologies such as energy, electronics, defense system, agriculture, and advanced manufacturing. Especially in BER science, recovering and extracting rare earth elements and critical minerals focuses on understanding their interaction with biological and environmental systems. So here I am showing some of the rare earth elements that are interested in the BER science. You can read through it. The next slide, please. And then here are some of the critical minerals that also relevant to BER. So what I am going to, next slide, please. So what I am going to show today is how we can use X-ray photoelectron spectroscopy, which is the surface-sensitive technique which can give elemental composition, oxidation state, and chemical bonding in this RE-CEM interaction with organic matter, microbial surfaces, and minerals in complex environmental system. So there are 4 applications I am going to show, 4 examples: oxidation state determination, RE absorption, microbial surface interaction, and characterization of biomineralization. Next slide, please. And so before I go into the examples, I will quickly go through the XPS basic principle. Next, please. So here, it's not working. Okay, go ahead. Next slide. Sorry, guys, for the thing. So basically, in XPS, you know that most of the electrons are orbiting in the different orbitals. So when you shine the X-rays on the material surface, it will have some energy, and that energy is given to the the orbital electrons, and then one of these electrons will kick off with the kinetic energy of Ek. So somehow if we can measure the kinetic energy, then if we know the incident energy of the X-ray, then we can calculate the binding energy using this particular equation. So the next one. So from this equation, we can identify the element present at any samples. Next one. So here I am showing the typical XPS spectrum from the silicon dioxide sample. You can see there are several peaks are shown here. Mainly you can see because of the different binding energy, each element will come into the different position. Silicon will come at one particular binding energy, carbon and an oxygen and things like that. Okay, next one. So as I mentioned, the XPS is a sensitive technique. We are getting the sampling depth about 3 to 10 nanometer. Next one, please. Next slide. Okay, it's not moving again. Next one, please. Sorry about this. Okay. So the main thing is about the XPS is the chemical shape. So what is the chemical shape is basically it's a change in binding energy of the core electron present because of the changing chemical environment. For example, if you take say aluminum in a metal, aluminum will have a nearest neighbors aluminum, but if you take the aluminum oxide, it will have a nearest neighbors as oxygen. So because of this changing environment, the binding energy of that particular aluminum 2p electron is going to be changed. So that's what we are going to measure in this particular chemical shift in the XPS. So basically, if you withdraw some of the valence electron, it is going to increase the binding energies like oxidation. If you give some additional valence electrons, that will decrease the binding energy, that is called reduction. So studying this thing, we can easily study the oxidation and the reduction of these elements. Next slide, please. So here is a typical carbon spectrum from the ethyl trifluoroacetate sample. You can see the carbon is in the 4 different chemical environment. In each one is going to give you 4 different peaks. So from these peaks, we can identify what are the carbon speciation is there. Next one. So here is a typical carbon spectrum from the soil sample. Again, you can see I have to fit this carbon spectrum with 5 different peaks. Each one will have a different oxidation state of carbon, which is linked to the different functional group. So again, by studying this different functional group, we can study about this organic material present in the carbon. So in XPS, we can always study about the metal or the oxides or the elemental composition and the bonding. At the same time, we can study the organics also. Next slide, please. Next one. So the first example I am going to show is the oxidation state determination of critical minerals. So here we can easily distinguish between cerium 3+ versus cerium 4+, and then uranium 4+ versus uranium 6+. Okay, next slide, please. So here I am showing 2 spectrum. The purple one is from the fully oxidized cerium oxide, which is in the cerium is in the 4+ state, and then in the red one is in the reduced one is cerium in the 3+ oxidation state. So you can clearly see these 2 spectra are completely different, and then you can monitor, especially if you look at the. Can you advance, please? If you can see this, the peak in the higher binding energies we call the satellite peak, that is completely absent in the cerium 3+ state. So by monitoring these 2 things, we can identify whether the sample is in the 4+ state or 3+ state or in the mixture of these states. By fitting these things, we can identify how much cerium we have in 4+ and how much cerium we have in 3+ if we have a mixed state. Okay, the next slide. Okay, go ahead. Next one, please. So here you can advance, Heather. So in this one, what we are trying to study is how the cerium nanoparticle is interacting with some organic compound like a ligand. So by studying the cerium nanoparticle is a little difficult because it is a combination of cerium 3+ and cerium 4+. So in order to study some basic understanding, we want to study in situ XPS measurement here. So what we did here is, the next slide please. we synthesize some model system of cerium-11 surface, which is fully oxidized, and then we annihilate that sample with a. You can go ahead. We anneal with a. We anneal with a. Anneal the sample in vacuum, and then we reduce the surface, and then we absorb this thing, the TMMA or the carboxylic acid, and then study how these things are interacting with cerium surfaces. Next slide, please. So here is a cerium 4 plus. Then when you anneal it, then it will become cerium 4 plus and cerium 3 plus, and then you basically deposit the carboxylic acid, and then you study the interaction using the XPS. Okay, go ahead. So here is the, you can advance, it will go fast through. So here is the typical XPS spectrum from the first one, the black one is from the fully stoichiometric surface. Then you can clearly see that is the spectrum I showed you earlier. And then when you have a reduce, you see you have a mixture of cerium 4 plus and cerium 3 plus. Okay, go ahead. And you can advance. Okay. So here we are again putting these carbonic acids on these two different surfaces. And then initially the green surface, it is a bare surface. There is nothing deposited. And then when we deposited the carbonic acid, you can clearly see there is a peak started coming at the 285 binding energy, which belongs to carbon-carbon or carbon-hydrogen bonding. And then the other one is coming around 289, which belongs to COOH bonds. But if you look at the reduced surface, you see that absorption is more. So for doing this kind of experiment, we can see the oxygen vacancies or the cerium 3 plus is promoting more absorption of these carbonic acid groups. Next slide, please. So here is I'm showing the oxygen 1s and the cerium 3D spectrum. Again, you can see that a black one is the bare surface and the red one is the absorbed surface. You can clearly see there is a peak coming. We have the C double bond OH corresponding. And then if you look at the cerium 3D, the cerium C3D spectrum also reduced a little bit. And then calculating, and then you can see there is a small shoulder coming on the lower binding energy side. So that means there is some cerium 3 plus is forming in the presence of this carbonic acid. So that is happening because some of the oxygens are transferring to the carbonic acid group. So that is called, we call this is dissociative absorption. If you look at the, go ahead, next one, please. If you look at the, reduced surface, you can clearly see the same difference, but the cerium 3 to cerium 4 is increases in this particular case. Because of this increase, we couldn't really calculate how much, what is the bonding nature in this particular case. The next one. So in the second one, I am going to show some example of XPS, how we can use in the REE absorption in mineral surfaces. Next slide, please. So in this particular work, what these scientists have done is they investigated the interaction of trivalent europium ions with the sodium montomonolite clay. So if you look at and then they use the XPS to study where these europium 3 ions are bonding in this particular mineral surface. So if you look at the structure of this montomonolite clay, it has three different features. One is called aluminum tetrahedral and octahedral silicon, and then you have the interlayer or the cation exchange in this layer here. So what is going to happen here is when you deposit this europium, it can go and then interact in these three different states. So they studied with the two different pH levels, low and high. Next slide, please. So here I'm showing the XPS from europium 3D in the montomonolite case, montomonolite minerals. When the pH is less, you can see it can only have a one peak at coming around 137.7 EV. But if you do the same experiment with a pH at greater than 6, you can see the peak is shifted, and then you have to use two peaks to fit this experimental data. So one is coming exactly at 137.7, but the other one is coming around 134 EV. So then what they decided, they did the same experiment on alumina and silica to mimic those two H states that I was mentioning. with a high pH state, and they found out that the alumina peak is coming around 134. The europium deposited on the alumina is giving the peak at 134. And then in the silica, it is also giving at 134, but it is also giving at that 136 EV. So based on these two experiments, what they found out is that in this, when the pH is high, the europium is binding in 3 different states, in the interlayered state and in the 2H state. But if it is the pH is 3, it is only binding into the interlayered state. It is not binding in the H state. Okay, so let's go to the next slide. We also did a similar study in the kaolinite. This is the lanthanum deposited on kaolinite, around 400 part per million lanthanum. Here they found out, and then they did some different lanthanum compounds, XPS, and then they compared with the actual data, and they found out that in this particular absorption, the lanthanum is bound to hydroxide surface, comparing the hydroxide XPS peak. It looks similar to the hydroxide peak. Okay, the next one. So now the third example I'm going to show is how the microbial interaction is going to affect this absorption of RE and critical minerals on the mineral surface. So as you know, the EPS, the extracellular polymeric substance, have a lot of functional groups that can influence these kind of interaction. Next slide, please. So here, what these scientists have done is they created different mineral surfaces. First is the bare mineral surface. They started with the goethite mineral surface. Then they did then they took the EPS, and then they mixed with the EPS and goethite, and then exposed all these surfaces to the arsenic 5 solution. And then they want to study how the arsenic is absorbing on this mineral surface. So if you look at the bare surface, you see there are 2 peaks in the XPS arsenic 3D spectrum. The blue curve belongs to arsenic in the 5+ oxidation state, and the blue one is in the green one is in the arsenic in the 3+ state. So as deposit arsenic in the bare surface, is already the surface is reducing some of the 5+ arsenics. If you look at the EPS, you can see again it is there's 2 peaks. But in this case, we have a more 3+ compared to the 5+. So the EPS functionalized group is giving more electrons to the arsenic 5, and then it is reducing the arsenic to 3+. But if you look at both EPS and the goethite surface, mineral surface to the arsenic, you see, if you look at the count rate, it is almost 30% less. So the less arsenic is bounding to this particular complex, but at the same time, it has more arsenic 3+. So as you can see, the EPS is doing some, EPS is influencing the absorption of these minerals in the metals in this mineral surfaces. They did a similar experiment in the other mineral surface called montomonolite. So here they found completely different arsenic interaction compared to the one that I showed you earlier. Here the arsenic peak is coming around 49 EV. So what they figure out from this experiment is that in this particular arsenic is bounding to the organic materials from the aromatic compound from this EPS. So here, basically this arsenic is in the organic, arsenic is bound to the organics. That's the reason it is forming into the higher binding energy. So you can also see, if you look at the carbon oneness, you can clearly see there is some extra peak coming at that particular binding energy. So as you can see, since I am almost out of time, as you can see, the XPS is very powerful technique to study all these kind of interaction. You can move to the next slides, to the final slides. Next one. You can skip this one. Yeah, leave it here. So this is our XPS setup. You can see that we can do some in situ measurements, like I showed you in the first example, and then we can do some doing all the kind of measurement in the inert environment, and then all these things what I mentioned we can study in our lab without any problem. So again, sorry for the fast, the short time I went fast. If you guys have any question, feel free to contact me directly or contact through the email. Thank you very much. Now we'll take over to the next talk.
Cool. Thank you, Shuttha. So I'm Will Q. I'm a staff scientist at EMSL, and I'm going to talk to you a little bit about high-resolution mass spectrometry for characterizing biological interactions with critical minerals. So without getting into too much of the weeds of how mass spectrometry works, it is important to introduce a few key concepts. So today I'm talking about high-res mass spec, specifically Fourier transform mass spectrometry. But like all types of MS, this is just a method to measure the mass-to-charge ratio of ions. Essentially, how much does a molecule weigh? And the extremely high mass resolution and mass accuracy of the high-res mass specs allows us to measure a molecular mass like 180.06339, and to that annotate a molecular formula like C6H12O6. And in any given spectrum, we can detect and annotate thousands of molecules, ranging from small molecules of 200 daltons up to big proteins of 30 kilodaltons or greater. So really, to show you an example of the difference between high and low resolution MS, we've got FTICR on the top, which is a very high resolution method, and a linear ion trap spectrum on the bottom. This is the same chemical, taurocolic acid, just analyzed by these two techniques. You can see in the left overview, you've got the same major features in both spectrum. However, the ICR spectrum is thousands of times more, the line widths are much narrower. And if you zoom in just to one of these isotope features, you see that in the ICR spectrum, we detect and resolve at least 3 different peaks, whereas in the ion trap, there's just one broad hump that means we're not measuring all of the features that are there, and we're not getting an accurate mass for those measurements. And beyond that, we even have the ability to get this really fine isotopic structure. So this is the, again, the same molecule, but just focusing in on species isotopologues that are only separated by 0.1 mass units. And you can see in the bottom is a theoretical simulation of that pattern, and on the top is what we measure. And you get a really high fidelity measurement, where we're able to detect and resolve things like a 34 sulfur substituted for a 32, or 2 carbon-13 substituted for 2 carbon-12s. And we get those resolved and with pretty good accuracy of relative abundances. And so that's the key to FTICR or high-res mass spec in general, is that we get accurate masses, we get accurate relative abundances, and we're able to resolve fine isotopes. Now, I've mentioned exact mass a few times. The key thing here is that all isotopes in the periodic table have a unique exact mass. That basically means the number after the decimal place. So carbon-12 is defined as exactly 12.0, but everything else has numbers after that value. And it's with those numbers, the fact that with these techniques like Fourier transform mass spec, we can measure 3, 4, or sometimes 5 decimal places accurately, that we're able to annotate molecular formula to a measured mass. Thinking especially about critical metals, the number of them have some very distinctive isotopic patterns. So, carbon as a nice example again, there's 2 main isotopes, carbon-12 and carbon-13. They have distinctive masses, and they have distinctive relative abundances, about 99% carbon-12, 1% carbon-13. Copper has 2 major isotopes as well. This time they're separated by approximately 2 mass units and have a different isotope ratio of 70 to 30%. Something like neodymium, on the other hand, has 7 naturally occurring isotopes, ranging from 5 to 27% relative abundance. Unfortunately, some elements only have 1 isotope, so aluminium or fluorine, for example, but they still have a distinctive mass we can measure. So as an example of this, a few years ago, one of my colleagues had synthesized a ruthenium catalyst complex and wanted to confirm the formula of that compound. So we took his sample and analyzed it on the 15 Tesla mass spec we have here in EMSL. And we could confirm here in black is the measured spectrum, and in red is the theoretical simulation for where the major isotope should be. And you can see we get a really good match. There's a few minor impurities present in the sample, but they're not an issue because the instrument has sufficient resolution. And you can see ruthenium has got isotopes from 96 to 104. But if we zoom in even more closely at any of those individual features, you can see that we get the ability to detect and resolve at any given nominal mass, both say 101 ruthenium substitution or 100 ruthenium carbon-13 or a 99 ruthenium and 2 carbon-13s. And we can measure these, and this gives us essentially absolute confidence that the molecular formula we've annotated is correct. Now, we have some unique capabilities at EMSL. One of those is our 21 Tesla FTICR mass spec. 21 Tesla is the highest field available for this type of mass spectrometry, which means it's capable of the highest resolution measurements. There's only two in the world, one here and one in Florida, and we've recently upgraded our instrument to have an Orbitrap FTMS on the front end. This gives us ability to do dual high-res mass spec measurements in one instrument at the same time. You might think, where would that come into play? And so as an example, one thing that's really important for our users is often things like microbial cultures. They want to know what metabolites are present in those samples. And we could do either imaging type analysis, or we could do liquid chromatography. And with this instrument, we're able to get both our intact molecular mass measurements, so the MS1 measurements, and our fragment ion measurements, our MS2 measurements, at the same time, but both in high resolution and with accurate masses, because we can parallelize our measurements between the two analyzers. And this allows us to do some really neat things. So in collaboration with René Boiteau at the University of Minnesota, we were looking for siderophores in fungal extracts. And so here we're looking for natural products that coordinate with iron. And so we ran the sample through a liquid chromatography separation, and we get a pretty rich chromatogram here, but we're able to search through every single mass spectrum looking for a very distinctive mass split. That is this 52.91147 that corresponds to the difference between a protonated ion and an iron-bound complex. We found a handful of those occurrences in this data set. We extract out that spectrum, and you see in a single spectrum, we get our distinctive cluster of peaks for an iron complex. We detected our protonated molecule as expected. We also observed an aluminium coordinating form of the same species, which we hadn't expected to see, which was pretty neat. So with this, we get a molecular formula, and we can confirm it again by zooming in and looking at the fine isotopic structure. In this particular case, we really pushed the instrument hard, and we're able to detect and resolve iron 56, 57, 58, oxygen, deuterium isotopologues, all in this very narrow mass area. This particular type of measurement is essentially only possible at 21 Tesla when you're trying to do things on a chromatographic timescale. Unfortunately, it's important to remember that formula isn't structure. So this is an example of 8 different structures that all have the same molecular formula. You can see there are some common motifs, like some of them have sugars and some of them are aromatic, but there are differences. And so if we actually want to know what the molecules are, we need extra information. And so that's where the fragmentation comes in. So at the same time that the ICR part of the instrument was getting the high resolution, accurate mass of the intact molecule, the Orbitrap part of the instrument was fragmenting that molecule and getting a high resolution spectrum of all of its fragment ions. And so this particular molecule produces hundreds of fragment ions. And thanks to our high resolution, we're able to detect and resolve multiple fragment ions per nominal mass and annotate to each of them molecular formulas. And you can imagine this is really important when you're trying to do some sort of structural identification or dereplication of natural products. And it's really important to note that this particular type of measurement in an untargeted fashion is just not really possible on any sort of commercial instrument you can get. And it's really one of the unique strengths we bring. Now, there are some other clever ways of doing these metal binding metabolomics. There's some work that came out of UCSD a couple of years ago where they showed you could do the same idea with having a chromatography up front coupled into mass spectrometer, but to make sure that you really probe all of the different types of metal or organic molecule interactions you can have, they had a secondary pump infusing metals after the separation. And what this allowed them to do was to essentially fish out which metabolites in their mixture can coordinate metals, and also, spike in different metals to see how those different binding efficiencies and coordination efficiencies change. Now, moving on a little bit from metabolomics into proteomics, we have this nice example from Jennifer Broadbelt's group a few years ago where they were looking at this SNase enzyme. And so on the top is just the spectrum that you get for it, noting that here we're studying what's called native proteins. So this is a protein that's sort of essentially in a biological conformation. It may be folded. If there's any protein-protein or protein-ligand interactions, those are preserved. And then they spiked in different metals, including lanthanum or neodymium, and they studied those complexes that formed. And so this is really useful for giving you, okay, what metals coordinate to my proteins, but they also use a technique called ultraviolet photodissociation, where they use a laser to break up these protein complexes and characterize the structure of them. And the way that this works is that you essentially blow up the molecule, the protein, when it is binding to the metal and when it isn't binding to the metal. And you get all of these pieces of the puzzle that tell you where the metal is coordinated to in the protein and how the presence of the metal may change the structure or the folding of that protein. So you can get some really useful insights. Again, this is a technique and a capability that we're able to do here at EMSL as well, thanks to some more unique instrumentation. We have got a Orbitrap instrument that is a high field Orbitrap, so a high resolution one, which we've custom modified in collaboration with Thermo to provide ultra high mass range. So a normal Orbitrap limits to about 6,000 mass to charge as the high mass. Here we can go to 80,000. We've also coupled it with a MALDI ionization source. So this allows us to do imaging mass spectrometry, and we've coupled it with a UVPD laser, ultraviolet photodissociation. And so this combination of techniques allows us to do native proteomics, studying these protein complexes, protein metal complexes, but also to analyze proteins directly from tissues using the MALDI source. And so mass spectrometry imaging, I've mentioned it a couple of times. There's a few ways to do this. The most common way people may have heard of is MALDI, which stands for matrix-assisted laser desorption ionization. And the idea here is, much like we've seen on the previous slides, you get a high-resolution mass spectrometry measurement, you get accurate masses, you can do molecular identification, but instead of getting it for a bulk sample, you get it spatially resolved. So in the case of MALDI, we have a laser that fires at a very specific position on a sample, maybe only a 15 micron area. It desorbs and ionizes molecules from that area, and we get a mass spectrum per position across the tissue. We repeat this measurement, moving the laser across the sample to sample the entire thing, which may be 10,000 pixels or more, and we can build up an image of what molecules are present in what positions on the tissues. We have an example on the top that was, using rat brain, and we were showing the really high resolution capabilities of our 21 Tesla. And in the bottom, we have examples of imaging proteins in biofilms. And this work was done on our UHMR instrument. And so we can do these measurements spatially resolved across a different range of spatial resolutions. We can look again at metabolites, at lipids, and at proteins directly from tissues, which can be very useful. And therefore, maybe there are ways to get spatial insights into how critical minerals are distributed across biological cultures and tissues. So the last thing I want to just highlight is it's really important to think about how you're doing your experiment before you get to the stage of sitting at the mass spectrometer. Obviously, you have your experiment design in terms of conditions, in terms of replicates, in terms of what you're studying. But it's also important to consider things like sample preparation. If you're doing imaging, are you sectioning it? If you're doing a bulk extraction, how are you lysing cells? What are you doing to extract it? And really the question is, how do we preserve these metal complexes if that's what we're trying to do? There are different ionization techniques to consider depending on the type of analyte you're looking for, its polarity, its mass, what charge states it might form, whether we want chromatography or imaging, or we're just doing direct infusion, and then which mass spectrometer we use. And then finally, the data analysis. If we're doing targeted analysis where you have putative identities you want us to confirm, or if it's an untargeted screening where you just want to know what's present in the sample, how does it interact with these critical materials? Obviously, we don't expect you to know the answer to all of these. These are just things to keep in mind when designing the experiment. Please reach out to us with any questions in that regard, and I'm happy to answer some questions. And now I'll hand over to David Hoyt, who's going to talk about magnetic resonance.
Thanks, Will. I don't know if anybody can hear me yet. Okay, yep. All right, so thanks. I think today's webinar is going to highlight a number of things as we have so far, the chemical and biological nature of molecules and how they might interact with metals and how it might help this area of research. NMR, I've had the pleasure of working with a number of you in the past. that might help identify small molecules from mixtures, as well as looking at structures, which is a strength of NMR, looking at bonding and chemical structures, as well as quantitation. And we'll talk a little bit more about those strengths in a minute. In earlier slides, it was wanted just again in terms of critical minerals and materials. There was a DOE 2023 report that's here and highlighting what's critical or near critical. And then there have been talks earlier where might we find these in various areas in higher amounts. Some of these are part of the reports and things that are there earlier. One of the things that I really wanted to get across was, in the past, we may have looked for molecules and profiling of what's there in soils or soil organic matter. We also look for things derived from biological or biologically inspired sources, whether that's plant or microbe. And a number of these things are metabolites or proteins, so larger biomolecules as well. And when we're looking for strategies and interactions of what's there and might be interacting more, we'll come back to this slide later, but essentially there are ways that bio-inspired systems need metals, a lot of these different metals, and will recruit and bring these in, whether that's a plant or a microbe, for their own use and when they're available. And even though some of these chelators may start out with a primary function to sense and bring in a particular metal, they've also been shown to bind other metals as well. In particular, some of the siderophores that might be primarily focused on bringing in iron also bind cobalt and nickel. And so how do we look at some of these things? And a lot of the strategies I think that'll be important are looking at what evidence do we have, not only on what is interacting, but how it's interacting, the chemical speciation and moieties. There are projects that we've been working with a number of you where the chelators, we see broadening and shifts, chemical shifts for organic acids, amines, etc. We'll come back to this slide later. Just some real basics. I'm going to keep it at a very high level here, but a lot of the nuclei that we look at, people might be most familiar with, have maybe some reasons for that, for higher sensitivity, proton or hydrogens, phosphorus, C13, N15, etc., and maybe even fluorine. We have probes that are highly sensitive for that on the liquid systems, especially. But I'm going to talk about, in general, magnetic resonance, both for liquid state, a little bit of solid state, and even how we use another magnetic resonance technique, EPR, when possible, so that we're characterizing nuclei when they're in a strong magnet and they orient. We can part and transmit radio frequency and how that nucleus either transfers it to other surrounding atoms or reads it back through the original. We wind up having measurements that then are highly specific to structure and the bonding environment around those molecules or the metals. So key features are it's non-destructive, and that's very important. A lot of times there can be less sample prep needed. It's non-destructive. Nothing in the measurement is changing the surface or consuming the material. So a lot of times it's a good consideration to think about doing this first in a screening or getting some of the key information from that and then handing off to a complementary technique or a downstream technique. It's isotope selective. It's highly quantitative, highly reproducible because we're not changing anything about the sample. In particular, there can be processes that we can follow. We'll come back to that later, but it provides a lot of structural information and also can provide other important information such as KDs or kinetic rates, correlation times, etc. And there are different approaches for whether you're trying to interrogate the metal directly or indirectly using a lot of information about ligand metal interactions. Some of the drawbacks are some of the nuclei are relatively insensitive. Oxidation state and magnetic properties can influence whether it's direct detection. Unfortunately, if something's problematic for direct detection from NMR, it doesn't mean it's not useful. There are other things that I'll talk about in a minute about paramagnetic shifts, etc. But it also means that if it's difficult for observation with NMR, it usually means that it's easy to detect and characterized by another magnetic resonance technique, also non-destructive, etc., EPR, or electron paramagnetic resonance. All right. This is a lot of information on here. I'll try to get at a higher level. This is mostly so that if we engage in what types of isotopes or how, what things we can tune to, this is a centerpiece for us to have discussions or figure out workflow. There's a lot of things that go into it. Again, if you want to look at that report yourself, there's a link here for the critical materials, the 2023 report. Again, a lot of times people are thinking about NMR with these particular stable isotopes. The periodic table has 118 elements, but there's over 130 NMR active stable isotopes. Not all of them are listed here. Another important definition, as we've heard, is the rare earth elements. That's essentially the 15 lanthanides and yttrium and scandium. Most of these have an NMR active isotope. We can talk in details about workflow strategies if we engage here in terms of the upcoming exploratory LOIs or as you get forward proposals and work strategies. And then even some of these things like we talked about in Will's talk where you can get exact masses and so forth that can help in identification or confirming or different strategies. As you couple these different techniques that we have at EMSL, again, structurally, getting structural information is a strength of NMR. The phase matters. A lot of these complexes, will remain soluble even with larger biomolecules like proteins, as well as metabolites or siderophores, etc. And if something's solid and it's on the surface or mineral surface or a powder or maybe on a membrane bound, solid state NMR comes into play there. And we'll talk about some advantages of some things that we've developed here, unique or, where we're the leaders in terms of looking at mixed phases of liquid solids, gases even, in terms of how we can bring magnetic resonance to bear. A lot of the things when we have these discussions are about oxidation state magnetic properties are specific for whether something's a good candidate for direct or indirect methods. But again, the strength is you can almost always do an indirect method and gain structural information about what's interacting with a metal. So again, if you're looking at direct detection, metal isotope free bound, the general observation is that you may see a change in the spectrum and a shift, broadening perhaps, and then that you can do this both if it's in the liquid state or the solid state. And then indirect methods of maybe looking at a ligand or a chelator candidate molecule. You may be able to do that from natural or maybe after fractionation, we can look at a whole series of metals that might be interacting with that chelator and learning something about selectivity and maybe on paths that we may all want to think about in terms of what are the important parameters that lead from a structure function relationship and understanding that help us ultimately to improve selectivity and possible deployment strategies in the end that will help for recruitment of rare metals that of interest. All right, the first example up here is copper. Copper 1 state, one of the isotopes that Will mentioned in 65. You can look at this by solid state NMR as we've done here. Copper is in complex by multiple ligands. This is dimethylthiourea. And although they all have dimethylthiourea in the three spectra, even the anions that it interacts with can affect what the spectra are, how you do fittings. There's a lot of computer simulations that go hand in hand with these fittings. This is an area in particular of the solid state NMR direct observation. When we do that, Andy Lipton is a good person with a lot of experience to look at the solid state for direct observation of metals. Same can be done for a lot of soluble metals, lanthanum, copper, lithium, and there's lots in the literature. I'm showing an example here of an indirect method where you're looking at the aromatics, aliphatics, some of the nitrogens that are involved in a complex, and even in processes where you go from monomers to dimers, you can use an indirect method like proton, and even something where you vary the temperature, you can restrict the number of structures that might be available. When it becomes more rigid or distinct, you'll get areas here where you see more detail, high resolution, couplings, etc., that all have a bearing on how this interaction is characterized, in this case in the aromatic region, and the aliphatic related to the methylenes that are part of this structure here. I'm going to keep moving. There's a lot to cover. Briefly did mention that as a number of things from way back when I was working with geochemists, looking at transformations of forsterite to magnesite, looking at silicon NMR and carbon NMR, we could see processes that needed to be monitored in situ. Some of these things were then looking at CO2 and liquids and solids all at one time. We had to develop rotor systems that then gave us the ability to look at mixed phases. These were also part of tech development LDRDs that I led, and with a design team of Eric Walter and Hardeep Mehta, Jesse Sears, and myself. There are a number of things that we've been able to look at over the years in terms of systems that you're trying to have a large range of temperatures and pressures and controlled atmospheres, and you can think of weathering and so forth, all sorts of things. This was a nickel aluminum solid, a liquid where it was isopropanol, and a gas, hydrogen, that we're controlling, and we had different pathways that we could look at in the terms of some conversions of, in this case, a benzyl phenyl ether, and confirming which pathways were actually in play. By having this where you have multiple phases all at the same time and possible control of that, and a wide range of temperatures and pressures, you can think of all sorts of things that you may want to follow. And we would have things that were both observable peaks that would be looking at things that were attributable to interactions on surfaces, as well as those that were still remaining soluble in the liquid. So I just wanted to point that out. I've talked about oxidation state and having different magnetic resonance methods. In particular, we took this a step further. This is something where Eric Walter and I worked together. And there's a holder that we had here that was actually able to be used both in the NMR and the EPR without unpacking. This was like a molecular sieves type of background, doped with copper. One of the oxidation states is EPR observable and difficult or impossible to do directly by NMR in the copper 2+ state, but it is NMR observable in the copper 1 state, as we saw earlier in the slide. In this case, we're doing aluminum NMR, which is more of the structure surrounding the molecular sieves. And what we found was in a redox reaction, and you can think of a lot of things in nature that would undergo redox. We started out with a strong signal for the copper 2+, and then saw a change in real time under the gas condition that we had. and the conversion, and then to copper +1. In this case, when we looked again, you can see now a sharpening of the features that you have from the aluminum MAS, and it would also be possible to look at the copper one directly as well. You can also see these are translucent and a little bit of a color change that's going on there as well for the copper that we had. All right, keep moving. We have just here as a, as a outlining, looking at indirect again, looking at something like EDTA or chelator of metals. You can have peaks that would be attributed to just the regions within the EDTA when they're free or uncomplexed versus, in this case, calcium complexed. And you can see the shift and the additional couplings that you're seeing in one of the resonances here, and what we're attributing to different parts of the molecule of the EDTA when it's complex. This was done as a function of different pHs, so again, environment matters. You can also, under the same environment, look and see a list of different metals. Here again, we have calcium free and bound, but magnesium, it's a different pattern and different amount of shift, and the amount of shift and so forth that you see, you can get in a series between the different metals highlighted here. Again, back to bio-based metal, chelators, etc. You have, in nature and a lot of these microbes, a quorum sensing type of arrangement where the availability of the metal, there's, once it's detected, there's more of chelators that are put out into the environment to help sequester, particular metal back into the cell. A lot of these are important to study further. There's different classes, and again, there would be labeling strategies, different things to think about. Again, I've looked at this with folks that are looking at chelators that and so forth that are available around glass that's going through dissolution with different metals that are coming out of the glass, etc. So these are all possibilities that I want to highlight in terms of strategies going forward. Love to engage with folks as they're putting together their proposals on what makes it easier to get structural information and these metal binding interactions from NMR. Okay, quickly, I'm going to try to do a few more things very quickly. We have just 3 examples where looking at a chelator that was involved in a breakdown of cellulose in a radioactive waste became more alkaline over time. This is some work that John Court did with isosaccharinic acid. It was able to bind actinides and unfortunately leach out. In this case, you might think about doing a surrogate of another lanthanide like europium-3, and here you're seeing the structure that we had initially, and then each of the progression of with the metal being found, the participation, and what happened in different parts of the molecule, and we can assign and map those out with high resolution. And also, this can be done in a protein environment. This work with Gary Butzko and Regina Trevino. Normally, of the protein-bound zinc, it was substituted with nickel. With a combination of doing computer simulations and machine learning, they did this whole series of site-directed mutants, some that bound or didn't bind the metal. There's different characteristics of the ones that did bind the metal. What are we learning in detail? How are we getting towards something that's more selective? And then finally, another protein that was derived from a microbial source that's able to bind a rare earth. When the protein itself is just enhanced by having an N15 label, you can do proton N15 experiments. They tend to cluster in NMR when they're unstructured, and then when it bound yttrium 3+, it became structured, and there's you can see the dispersion of signal. I don't have a lot more time to talk about, but anyway, this is some work a colleague of mine, Emily Close, was involved in. And again, the whole series of capabilities that you've heard about in the June 11th and today. Today we had the features of these ones in yellow that were discussed in the talk earlier. As you're putting together things with your exploratory proposal interest, you may think about incorporating a suite of these. All of these are available at EMSL, and I thank you for your attention and any questions you might have. These are some of the collaborators and people in the group that are involved in doing in situ NMR or solid state EPR, like Eric, or a number of folks that contributed, again, John, Gary, Emily, for some of the slides and examples that they gave me today. Thank you for your attention.
Thanks, David. I'm going to have our speakers come back on screen so that we can answer a few questions. And we will try and stay a little bit past 1 o'clock if you have additional questions so that we can get those answered. So first question is for Shuttha. Shuttha, can X-ray photoelectron X-ray photoelectron spectroscopy distinguish between different types of organic functional groups involved in rare earth elements on critical mineral sorption?
Can you see me? Okay, yes, yeah, definitely. I already mentioned, right, in a couple of my slides, the initial slide. So we can easily see the carbon speciation, and then we can also see the oxygen speciation. So each organic thing is going to have a different binding energy. So we can easily, distinguish between the, say, if you have a lanthanum, some organics compound, we can clearly see the differences. No problem at all.
Okay, thank you, Shuttha. Will, a question for you. Can you really detect metal organic complexes directly from complex mixtures like soil, microbial cultures, or plant extracts, or do the compounds need to be purified?
That's a great question. So the answer is we can analyze these mixtures, excuse me, thanks to the high resolution of our capabilities. The challenge is, of course, are those complexes stable in solution? So you might have some mineral metabolite interaction, you might have some metabolite coordinating a metal, and when it's in a biological system, maybe that's okay. But once you extract it into an organic solvent like methanol that we use for the mass spectrometry, maybe that dissociates that coordination. So there are challenges to those measurements, and it needs to be kept in mind. However, there is this whole area of native mass spectrometry, which is all based around the idea of making these measurements of the complexes as they are in biological systems.
Thanks, Will. Question, is gallium in the critical minerals list that would be compliant with EMSL research? This is a question for any speaker.
Yes, gallium is listed in the critical minerals table.
Okay, perfect. Another question. They said, hi, thanks for the great presentations. For the identification, and I'm probably going to butcher this, siderophore. Yes, oops, it disappeared. Hold on. Yeah, hold on. Oh, again, I lost it. Shoot.
It's in the answer tab. So the question is,
okay, so do we need to use the solid CAS agar assay or the liquid CAS assay?
I have to be honest, if this I'm assuming this question is for me. I do not understand the question. I'm not familiar with the CAS agar assays.
Okay.
The way that these experiments were conducted with the fungal siderophores was my collaborator cultured the fungi, presumably on, agar, as you're saying. They conducted the extraction, which could be as simple as just taking that biomass of fungi and extracting it with methanol or acetonitrile, shaking it, maybe using a sonicator to break up those cells, maybe using a bead beater to break up those cells.
Okay. All right. Thanks. That was a good answer. You did provide a lot of context. Thanks. So Dave, how does NMR distinguish between metal-bound and free organic ligands like siderophores or organic acids?
So there are structural assignments that are made to these, and they're either in libraries or we do. It may be beneficial not to look at it in a mixture if you have a particular target. maybe doing some sort of fractionation if that's a bench experiment that you want to do. But you can do it for mixtures and look for these things. And again, as I kind of alluded to in one of the slides, where you can have what would be normal and free and metal in the metal not complex, and then you're going to look for shifts and broadening and so forth as evidence for interaction. And then again, you may want to go further from that. If you can get isolation and then look at a series of metals, you may get a lot more insight as well.
Thank you. Okay, another question. Will NMR identify, I'm trying to think here. Is it
Vanadium.
Okay. Do you want to just take it?
I'll ask the question to David.
Okay, yes, please do.
David, will NMR identify vanadium-3 from vanadium-4 in 1,000 part per million level samples? Or can vanadium-4...
That's a good question that I'd have to drill down into, to be honest. I'm trying to be very honest about this. I think why I put those things of all the different NMR nuclei and so forth is some of these things are details to the point of, with a little bit of background, I would be able to guide you better on a question like that.
Okay, thank you for that. Another question. Does the
research identify 3 and 5?
Does the research question have to do with how a biomolecule complex or how a biomolecule complexes with a critical element? In other words, how does an organism sequester critical metals?
I'll take a stab at that. So there are papers out there when you have microbes in the presence of what's available beyond what I was sort of alluding to, where it's sequestering or sensing what's around. If you have additional metals that are not there, you're essentially applying a selection pressure. And so there can be responses, and there are things in the literature where microbes will potentially change or alter biosynthetically something that's available and interacting.
Okay, thank you.
Just to expand a little bit, I think the other half of this is, how are we phrasing these research questions for EMSL's proposals? So you might come to us with a question of, I'm really interested in microbial bioleaching of critical minerals from mine tailings. And so what we can try and help you with is, okay, what are the biochemical processes by which that happens? And so your hypothesis may be driven more by an application, and we can help a little bit with the fundamental science underlying that.
Right. And I think that leads into, we have an open call right now, the EMSL Exploratory Research Call. The letters of intent are due on July 2nd. So it's a great time to be reaching out to our speakers here, to our integrated research platform leaders, to ask questions that can help you with a successful proposal when you're writing it. So we would all encourage you to review the exploratory research call content on our website, to reach out and ask questions about which resources and instrumentation would be right for your particular proposal. And we will have this webinar recording on our website for you to go back and watch within about a week. We also invite you to go back and look at the June 11th webinar, where we also talked about critical minerals research and our resources. Additionally, we had an overwhelming response for a desire to have a copy of the slides from today's presentation, and we will make sure that those are provided to our attendees. So with that, we want to thank you for attending today. Thanks for your questions. Thank you to our speakers. And we hope that we will see you at EMSL Summer School, July 7th through the 11th. It is free, and you can watch right from your computer at home or at your office. So thank you for attending today. Bye-bye.